A therapeutic monoclonal antibody is a massive, intricate physical structure composed of precisely folded polypeptide chains and held together by exact disulfide bonds. But if we zoom in on the constant heavy 2 domain, the CH2 region located on the tail of the antibody, we find a specific nitrogen atom on an amino acid called asparagine 297. Attached to this exact residue is a highly complex branched carbohydrate chain called a glycan. Noding this specific sugar chain correctly is one of the most difficult manufacturing bottlenecks in modern pharmacology. In biopharmaceutical manufacturing, safety and efficacy are tracked using critical quality attributes, or CQAs. These are strict physical, chemical, and biological limits that a drug must meet to achieve regulatory approval. For a biologic, glycosylation is a primary CQA. The exact arrangement of the sugar chain dictates how the drug behaves in the human body. Small variances in microheterogeneity, like the specific degree of branching or the presence of a single fucose molecule, drastically alter the drug's pharmacodynamics. If the cell attaches the wrong terminal sugar, the patient's immune system might flag the drug as a foreign invader and attack it. If the glycan is trimmed improperly, hepatocyte receptors in the liver will bind to the antibody and clear it from the bloodstream before it can reach the target tumor. The industry relies heavily on Chinese hamster ovary, or CHO cells, to mass produce these therapeutics. However, a native hamster cell is optimized for its survival, not for synthesizing optimized human immune proteins. To reliably match the rigorous CQAs of a human biologic, engineers have to aggressively reprogram the cell's metabolism. To understand why glycosylation is so difficult to control, we have to look at how proteins are built. Polypeptide synthesis is strictly templated. A ribosome reads a rigid mRNA sequence and links amino acids in an exact, deterministic order. But glycosylation does not work this way. It is a non-templated, highly variable metabolic network. The glycan is constructed step by step as the protein moves through multiple compartments of the endoplasmic reticulum and the Golgi apparatus. There is no genetic blueprint for the final sugar structure. The composition of the glycan is dictated entirely by which specific enzymes and nucleotide sugar substrates happen to be physically present at the exact moment the protein passes through a given organelle. This means transit time is a critical variable. If the protein moves too quickly through a Golgi compartment, the local enzymes will not have enough time to finish their attachments, resulting in an incomplete, highly heterogeneous glycan. To solve this inherent biochemical variability and force the cell to produce a uniform product, bioengineers deploy a dual-vector control strategy. The first vector is permanent genetic hardwiring. Using CRISPR-Cas9, scientists edit the CHO cell genome to overexpress desired human glycosyl transferases, or entirely knock out the host's native competing enzymes. The second vector is dynamic bioprocess tuning. Engineers actively manipulate the chemical environment of the bioreactor, adjusting pH, shifting temperature, and timing specific nutrient feeds to throttle the speed of the cellular assembly line. To manufacture predictable, clinical-grade biosimilars, we must map out this entire biological assembly line, identify failure points, and hijack the process from end to end. This diagram maps the glycosylation pathway. At the EL membrane, an enzyme attaches a massive 14-sugar precursor to the protein's asparagine site. The cell immediately trims specific glucose and mannose residues, folding the glycoprotein. Once passing quality checks, it translocates into the cis-Golgi. If this early trimming process is incomplete, due to high processing speeds or limited enzyme availability, the protein exits the cis-Golgi retaining too many mannose sugars. This creates a high mannose glycan. High mannose structures are a major clinical liability. While they sometimes bind effectively to target cells, clearance receptors rapidly identify them in the human body, drastically increasing the drug's clearance rate and destroying its therapeutic half-life. Engineers can actively disrupt this stage in the bioreactor. By intentionally limiting glucose feeds or applying specific alpha-mannositis inhibitors like DMJ, they purposefully stall the trimming phase to evaluate these high mannose variants. Precise trimming in the ER and cis-Golgi is a non-negotiable foundation. If the base structure isn't stripped down to its correct core, the cell cannot build any of the complex modifications required downstream. Transiting into the medial Golgi, the protein encounters the GNT enzyme family, the architects of branching. GNT1 and 2 construct a standard biantennary structure. Expansion enzymes, GNT4 and 5, add molecules at different angles, pushing the architecture into wider triantennary and tetraantennary branches. This is a prime target for metabolic engineering. By genetically altering the CHO cell to overexpress GNT4 and 5, engineers force the Golgi apparatus to build higher branched structures. The geometric purpose here is structural capacity. Every new branch acts as an additional physical anchor point for terminal sialic acid, which will be added at the very end of the pathway. However, another enzyme, GNT3, can add a bisecting glucose NAC directly into the middle of the core. This addition physically blocks subsequent enzymes like galactotransferase from accessing the glycan, abruptly halting any further modifications. Because branching dictates how many protective sugars can be attached later, the reactions occurring here in the medial Golgi set the absolute structural ceiling for the molecule's eventual circulatory survival. Moving into the trans-Golgi network, the glycan encounters a highly specific enzyme known as fucosyl-transferase 8, or FUT8. The sole function of FUT8 is to attach a core fucose residue to the exact GLAC molecule situated right next to the asparagine root. This microscopic physical addition has a massive clinical consequence, particularly for IgG antibodies designed for cancer therapy. When an antibody binds to a tumor cell, its Fc tail needs to recruit the patient's immune system to destroy it. But the presence of that single core fucose creates steric hindrance, physically blocking the antibody from binding tightly to the Fc gamma R3A receptors on the patient's natural killer cells. The result is a severe systemic failure. The antibody finds the cancer, but cannot trigger antibody-dependent cellular cytotoxicity. The natural killer cell fails to engage, and the tumor survives. To solve this, bioengineers deploy a permanent CRISPR intervention. They delete the FUT8 gene entirely, or knock out the specific transporter that brings GDP fucose into the Golgi. The assembly line is forced to run without the fucose pathway. Removing that single sugar molecule yields a staggering clinical result. Non-fucosylated therapeutic antibodies demonstrate up to a 100-fold increase in cellular cytotoxicity against cancer cells. You cannot achieve complete afucosylation reliably just by tweaking bioreactor nutrients. When a specific molecular structure actively harms the drug's efficacy, permanent genetic deletion is vastly superior to dynamic process tuning. In the final stages of processing, galactosyltransferase steps in to extend the glycan branches with galactose. Immediately following this, sialyltransferase attempts to cap those exposed ends with a negatively charged sialic acid molecule. Here, a critical species mismatch occurs. Human glycoproteins utilize an enzyme called alpha-2,6-ceatat to create highly stable linkages. Native CHO cells lack this gene entirely. They only possess alpha-2,3-ceatat. Furthermore, the sialic acid precursor that CHO cells synthesize, Nu-5GC, is highly immunogenic in humans. Our bodies naturally produce antibodies against Nu-5GC, meaning a native CHO glycan can trigger an aggressive inflammatory response known as xenosylitis. CRISPR genome editing knocks out the native hamster enzymes and knocks in the human ST6-gal1 gene, forcing the cell to form humanized alpha-2,6 linkages. To provide raw material, overexpressing CMP sap transporters pushes a massive flux of sialic acid into the Golgi, fueling the new enzyme. This genetic foundation is maximized through bioprocess levers. Shifting the bioreactor pH down to 6.85, bolus feeding the culture with manganese, and supplementing with uridine provides the chemical catalysts needed to drive galactosylation and sialation to their theoretical maximums. Proper sialic acid capping serves as the biologics' ultimate invisibility cloak. It masks the underlying sugars from the liver's clearance receptors, ensuring the therapeutic protein remains circulating in the bloodstream long enough to do its job. Zooming out, these engineered cells execute millions of simultaneous reactions inside a high-density bioreactor. This requires absolute synergy. Genetic knockouts establish structural limits, but the kinetic flux, The actual speed of the assembly line is driven entirely by environmental process parameters. A perfect genetic edit can still yield a failed drug. If engineers feed the culture too much glutamine, the resulting ammonia accumulation inside the reactor will severely suppress silation, overriding the cell's engineered genome. To prevent the cell from pushing proteins through the Golgi too fast, operators will execute deliberate temperature shifts, dropping the culture from 37 to 30 degrees Celsius. This purposely slows cell division, giving the Golgi enzymes the processing time they need to complete complex glycans. Real-time continuous monitoring of pH, dissolved oxygen tension, and osmolarity is required to keep these engineered metabolic pathways operating efficiently without triggering cellular stress responses. Successful biomanufacturing at an industrial scale demands absolute, precise alignment. The engineered host genome must be perfectly calibrated against its dynamic chemical environment. The industry has largely mastered the production of standard IgG monoclonal antibodies. But in the context of modern pharmacology, a standard biantennary antibody is relatively simple. We are entering a new wave of hyper-complex therapeutics, including intricate Fc fusion proteins and advanced bispecific antibodies, and bioengineered glycosaminoglycans like heparin, molecules requiring a biosynthetic pathway of over 20 different enzymes to construct. These novel structures will severely strain the natural post-translational limits of our standard CHO cell platforms. Moving forward, manufacturing will require pushing glycoengineering well beyond current capabilities. It will necessitate the design of entirely new host organisms and highly advanced bioprocess models capable of sustaining them. We have proven that the chaotic, non-templated nature of the Golgi apparatus can be tamed. Completely mastering that intricate glycan code is the ultimate non-negotiable prerequisite for the future of synthetic biology.